The image on the screen shows the consequences of eutrophication. Eutrophication is a result of an excess of nutrients in treated sewage water, which is discharged into water bodies, resulting in the formation of extensive mats of floating plants such as algae blooms. These nutrients are nitrogen and phosphorus, primarily as ammonium and phosphate. Eutrophication not only represents a threat to the ecosystem, but also to human health. This is an urgent problem that needs to be addressed. That is why we investigated a non-biological option for ammonium and phosphate removal from real black water using silicate-based materials. Nutrient concentration in on-site wastewater treatment systems, effluents need to be reduced for safe discharge and/or for water reuse. The standards for water discharge require 70% removal of the total nitrogen, 80% removal of the total phosphorus, and for the pH of the effluent to be between 6 and 9. We investigated the following silicate-based materials: polonite, which sources phosphate, and clinoptolite, which targets ammonium. All water samples were obtained from a single-pass on-site water treatment system, and all nutrient characterization was done using commercially available kits. The system counts with the following steps: the raw black water influent, where black water refers to the wastewater from toilets containing urine and suspended solids. The influent goes through an ultrafiltration column which removes the suspended solids, then through a granulated activated carbon column which removes the soluble organics. As the final step, an electrochemical cell for disinfection. It is important to find the best place in the system where to include the nutrient removal step. For that reason, we tested clinoptolite and polonite using water samples after each treatment step of the system. As shown in the graph to the left, clinoptolite performance is independent of the water type being used. On the other hand, polonite's performance is improved if the water type used has already gone through the gag column step, shown and represented by the pink triangles. Keeping that in mind, to improve polonite sorption capacity, the nutrient removal tanks were installed after the gag column step and before the electrochemical cell, as shown in the schematic, and as well shown in a real picture of our system. Now, the question becomes, are these materials effective in a single-pass on-site water treatment system? In this graph, we have the effluent pH on top, the ammonia removal percentage in the middle, and the phosphate removal percentage in the bottom. Inside the graph, the gray shaded area shows the data collected before the nutrient removal tanks were attached to the system, and the green shaded areas represent where the values will ideally be within if they were to a standard. What we can clearly see is that the nutrient removal is immediate upon the installation of the tanks. Comparing what we have inside the gray shaded area to what comes after the tanks have been installed. The clinoptolite removes more than 70% of the ammonia. With this percentage, clinoptolite reaches standard. Polonite removes more than 50% of the phosphate. Unfortunately, this is below the required 80%. And the pH is also too basic due to the presence of polonite as one of our minerals. Not only these minerals are effective in an on-site wastewater treatment system, but also clinoptolite can be regenerated using hydrochloric acid, and polonite can be regenerated using potassium hydroxide. In the graph in the left, the pink triangles show the clinoptolite after regeneration with hydrochloric acid can solve the same amount of ammonia. This is the same case for polonite. The plot in the right, with the blue triangles, show the polonite sorption of phosphate remains the same after regeneration with the potassium hydroxide. Hydrochloric acid and potassium hydroxide were chosen over some other regeneration solutions shown for both materials in the plots, such as sodium chloride and sodium hydroxide, as the concentration of sodium should be minimized for water reuse. Having chosen the regeneration solutions, we investigated how many times could the minerals be regenerated using the same regeneration solution for each cycle. Ammonia and phosphate removal was successful after multiple regeneration cycles. The graph on the right shows the ammonia sorbed and desorbed from clinoptolite. Hydrochloric acid can desorb 70 to 100% of the ammonia from the clinoptolite for up to 7 cycles. The graph on the left shows the phosphate sorbed and desorbed from polonite. Potassium hydroxide can desorb less than 50% of the phosphate sorbed on the polonite. This is most likely due to the phosphate precipitation. In this study, we were able to answer the following questions. Where in the system should the sorbent materials be incorporated? After the gag column and before the electrochemical cell. What are the sorption capacities of clinoptolite and polonite for use with real black water? Clinoptolite 13 mg of ammonia per gram and polonite 4 mg of phosphate per gram. Are both materials effective in an on-site wastewater treatment system? Yes, they work immediately after implementation. Something to be aware of is that the high pH of the effluent might be a limiting factor. Can they be regenerated for multiple use cycles? Yes, clinoptolite can be regenerated using hypochlorite acid to dissolve 70 to 100% of the ammonia, and polonite can be regenerated using potassium hydroxide, which dissolves less than 50% of the phosphate. Therefore, the silicates minerals are a promising non-biological option to remove the nutrients causing eutrophication.